Hello everyone, welcome to the channel. Today we're going to talk on the topic what is DNA. Today we are diving into the blueprint of life itself, DNA, which stands for deoxyribonucleic acid. Imagine it as an instruction manual for building, maintaining and operating every living thing. And guess what? This magic happens right inside the nucleus of your cells. Ready to unravel the secrets? Let's go. This is an example of a double helix of the DNA condensed in the chromosome depicting the nucleotide base pairs and sugar phosphate backbone. You ever seen a twisted ladder? That's what a DNA looks like. Picture two long strands made of sugar and phosphate holding together rings of nitrogenous bases. We have got adenine, thymine, cytosine and guanine forming our rings. Think of them as the ultimate friendship bracelet. A always pairs with T and C always pairs with G. Friendship goals, right? Alright. Time for some matchmaking. Adenine and thymine are like Romeo and Juliet, always pairing up. And cytosine and guanine, G, they are like the power couple, always together too. These pairs are held by hydrogen bonds, kind of like tiny magnets keeping our DNA stable. Let's give a shout out to these dynamic duos. And this figure also has an example of central dogma of molecular biology. DNA is converted to mRNA by a process called transcription, and mRNA is converted to protein by a process called translation, which is a key pathway of all human beings. Imagine you had a magical photocopier that would copy an entire instruction manual perfectly every time. That's DNA replication. First, the DNA unwinds like a zipper. Then new nucleotides come in and pair up with the original strands, making two identical molecules. It does involve a lot of enzymes such as primase, polymerase, RNA primer, and ligase. But in short, you get two daughter identical copies of DNA. Magic, right? So DNA is amazing. How does it fit into our cells? It's all about organization. Genes are like individual recipes in a cookbook, telling the body how to make proteins. These genes are packed into chromosomes, 23 pairs of them in humans. Think of chromosomes as a super organized filing cabinet storing all your genetic information. Neat, huh? So this example shows how DNA is condensely packed inside the nucleus of a cell. Time for some molecular magic. DNA first gets transcribed into mRNA in the nucleus. Then this mRNA travels to the ribosome, which is like a kitchen where it translated into proteins. Each set of three nucleotides in mRNA is called codon. It's like a recipe ingredient telling the ribosomes which amino acid to add next. So in this figure, you can see how RNA polymerase is using the promoter region of DNA in creating a newly synthesized RNA strand. Ready for some drama? Mutations are changes in the DNA sequence. They can be tiny like a single nucleotide change, which is point mutation. Some mutations are harmless, some are superpowers and others they're a bit troublesome. It's a nature's way of mixing things up. Here we can see some examples of mutations. Silent, which is typically harmless, changing one codon at a time in creating the same amino acids, which doesn't have any significant effect on the gene. On the other hand, nonsense had a mutation which ultimately stops the protein translation. It might have harmful effects in living beings. And conservative and non-conservative has either beneficial or harmful effect. Time to channel your inner directive. DNA plays a starring role in forensics. Ever heard of DNA fingerprinting? It's like a high-tech magnifying glass for crimes in investigations. At first, detectives collect samples such as blood, hair, saliva, you name it. Then, through a process called PCR amplification, they make a tons of DNA copies. Finally, gel electrophoresis separates DNA fragments by size. It's like solving a puzzle with invisible pieces. And yes, just as cool as it sounds. As you've seen in this figure, the crime scene sample matches closely with the suspect too. Depending on the band size and band width, we can conclude the suspect too is a possible suspect of crime scene sample. Here's where science meets sci-fi. In biotechnology, DNA is like a magical tool. Think genetic engineering, modifying an organism's DNA to achieve desired traits. Enter CRISPR-Cas9 system, our precision tool for DNA editing. From creating drought-resistant crops to developing gene therapies, the possibilities are endless here. It's like having a superpower, but for real. So this example is a simple explanation of how CRISPR-Cas9 system looks like, involving a Cas9 enzyme complex, how it attacks the DNA at a precise point. Hold on to your hats because the future is mind blowing. The Human Genome Project mapped all our genes, paving the way for personalized medicine, tailoring treatments to our unique genetic makeup. Ongoing research might even cure genetic diseases and unlock secrets of evolution. Imagine living in a world where your doctor's advice is as unique as your DNA. We are on the brink of something huge. Time for some history lesson, but don't worry, this one is exciting. In 1869, Frederick Miescher discovered nuclein, now called as DNA, in white blood cells. Fast forward to 1950 and Erwin Chargaff unveils the base pairing leaves. A has double bonds with T and the C has triple bonds with G. Rosalind Franklin X-ray diffraction images in 1952 provided crucial clues, and in 1953 Watson and Crick revealed the iconic double helix structure. It's like a detective story but with scientists as heroes. Continuing our historical journey, each step brings us closer to understanding like itself. From Meisher's initial discovery to Chargaff rule and Franklin's pivotal images and Watson-Crick Eureka moment, each scientist added a piece to the puzzle. It's a testament to human curiosity and perseverance. Who know? Who knew our genetic code has such a dramatic backstory? Ready for some jaw-dropping fun facts? If you stretched out the DNA in all your cells, it would reach the sun and back multiple times. Except for identical twins, no two people have the exact same DNA. And get this, DNA can store digital information too. 1 gram can hold about 700 terabytes of data. Plus, the fruits and veggies we eat contain DNA. Next time you munch on an apple, remember you're consuming some genetic info too. Thanks for watching. I hope you guys learned something today. See you soon. Thanks.